Hello, and welcome to our poster about clusters of interactions between operational taxonomical units in human gut microbiome. Interactions within human microbiota are not yet fully understood, but they are already being exploited in treating various diseases, either directly by fecal transplant or indirectly by, for example, change of the diet. The second one, change of diet, is often used to avoid dysbiosis. Even though we don't necessarily mean what exactly is the consequence of dysbiosis, we are able to say that it is not healthy. But over 50% of the microorganisms living in human microbiome functions that are, well, not yet understood. One of the hypotheses why it is so difficult to analyze the human gut microbiota is that it is extremely flexible, because it is not important which taxon exactly is present in the microbiome or what functions do the specific microorganisms serve. The hypothesis says that what is important are the functional communities that are formed by the microorganisms, which are together serving the purpose that is larger than the single ones of them. In order to analyze this, we used the data from American Gut Project. We downloaded it using the Cheetah software and preprocessed it using a deep layer pipeline, trimming the sequences at 150 base pairs and discarding all shorters. We also removed samples other than the ones marked as a stool and removed the bloom sequences. Then finally we rarefied them to 10,000 reads, yielding 18,000 samples that were eligible for further analysis. The analysis consisted of using the multidimensional feature selection algorithm to extract the information about interaction between the taxons. This way, we obtained the matrix of the interactions and feature importance, which was able to tell us for each column meant which taxons we can use to predict the presence of the specific taxon. Then, to reduce the amount of noise in the matrix, we applied the weighted random walk algorithm. So we treated it as a graph of interactions, with each taxon being a vertex and interaction between two taxons being an edge. So we used the weighted random walk on the directed graph, and the probability of choosing a specific taxon to be a part of our new subgraph was inversely proportional to the degree of the node. So this way we were able to avoid the so-called hubs, the taxons which were connected to many other nodes which aren't necessarily important to our analysis at the moment. So the taxons from our new graph were then clustered using the k-means algorithm, and we analyzed their functions using the PyCrest2 software. Finally, to validate our hypothesis, we trained a set of XGBoost classifiers thus proving that it is possible to use the functional annotations of the cluster or of taxon to specify whether it will belong to a specific cluster of interactions, even though there is not much significant overlap between the functions of the organisms belonging to either to the specific cluster or out of it. What you can see on the bottom left is the visualization of one of the graphs that we were analyzing. If you were interested in this study, we are looking for a PhD student, both for this and for other similar research. Thank you for your attention.